Thank you so much for the nice introduction. Hello, everyone. I'm Songpeng Zu. I'm now a postdoc in Professor Bing's lab. And today I'm very glad to introduce our recent work about comprehensive single-cell analysis of chromatin accessibility in the adult mouse brain. So it's about more than 100 years ago that Kagel, using the Gardner method, to discover that our brain system are organized actually by the individual cells. And so currently we still, the brain system is quite important for us. They control how we think, how we drink coffee, or even how I do the presentation today. And researchers are very interested in understanding what kind of cell types we have in our brain system. And the Brain Initiative was launched in 2013, which trying to do the brain research study using the advanced innovative technologies. And BICCN is the Brain Initiative with the full name of the Cell Census Network. The basic aim for the BICCN is trying to identify the cell types and based on different kind of measurement, such as transcriptomics, epigenomics, and spatial transcriptomics, and other measurements. And this project was launched in 2017. And here you can see we have different modalities and based on the different and have the corresponding technologies. So today my topic will focus on the attack state analysis, which is actually on the epigenomics part to identify the cell types in the brain system. Oh, by the way, so feel free to stop me at any time if you have any questions, want to add comments, or even correct me. And so our aim using the attack data to identify the structural element, and they are critical important that control the cell type specific behaviors. So here is a figure introduce the nuclear structure, and the chromatin in a large scale have some kind of territories, and and also they have another level structure kind of A compartment or B compartment. Now here B actually corresponding to red part here, which are heterochromatin regions. You can see typically there are no RNA data there, and also you cannot see the chromatin accessibility in those regions. And for there another compartment is A compartment. You can see the corresponding open state and the signals. And the important part to identify as a marker for the A or B compartment is the H3K9 trimethylation. And if we go further to another level of this part, so we can see we have this kind of active compartment or inactive compartment. So cis-regular elements are those DNA elements in the genome that can be enhancer or repressor or the insulator or the promoters. So typically, for example, for enhancer, they are bound by the transcription factors and can regulate the downstream target genes. And here is another figure to introduce the detail of this part. So typically in promoter region, we have portal bonding, and transcription factor will bind into cis-regular elements like enhancer and promote corresponding transcription of the downstream genes. And in the meanwhile, there are also some histone modification or demethylation to identify the corresponding classes of these different cis-regular elements, which we call CRE. So for example, for chromatin accessibility, we can identify either promoter, enhancer, insulator, and so on and so forth. And for H3K27 acetation, we can also mark the potential promoter region, enhancer region. And we can also have other parts, for example, the CT7 marking the corresponding insulator regions. So today our topic will focus on the chromatin accessibility to identify all the potential candidate cis-regular elements, which we call CCREs. And in the meanwhile, recent JIVA study also showed that more than 90% of disease-associated variants actually map outside of the promoting coding sequences. And a substantial ratio of those variants actually located in the cis-regular elements. And if we have the single cell data, we can actually identify different cell type and even subtype so that we can more accurately mapping those variants to the particular cell type and the corresponding cis-regulated elements. Here is another, so in our paper, we mainly works on the mouse model, which is an experiment, the most widely used animal model and have a critical role in the investigation of gene function in vivo. So here is the work from Yang in our lab that he used human cerebral region and mouse cerebral region to do the integration. And based on the motif part signal, they can integrate well. And you can see he also the human cis-regulated elements identifying human brain to the mouse genome. And then, so you can see large percent of those cis-regulated are conserved in our mouse genome. And a further CLIVA study for the neurological diseases also noted that those kind of disease-related variants are mainly located in the non-coding region and especially for the conservative regions. So before I go into my work today, I would first acknowledge all the members joined in this program. So this is a very big program, and I'm very lucky to be in Professor Bing's lab, and he introduced me to join in. And Yang and Conley are also the co-first author in this paper. So our data is mainly generated by the Center for Epigenomics, which at that time were the lead by Sebastian, and we use the Sky ATAC for the sequencing for our sequencing. And also we have our collaborator Joe Ecker group. They can share their they also working on the epigenomic part, but mainly focus on the single nuclear methylation data set. And all the examples, mouse brain examples are dissected from MAGA team. And we also get lots of help from the Allen Institute. They can provide their single cell RNA data for us to do the annotation, and they also review our paper. So our work is kind of an extension towards our previous work in the adult mouse cerebrum regions. So this also the first step for the BICCN, which I think was launched, published the kind of a result from different group in 2021. So at that time, different groups focus on the several regions, and in our group, we focus on the ATAC data and do the dissection for the mouse brain in these regions. And we have about 800,000 cells at that time, and we identify about 160 cell types. You can see our GABAergic glutamatergic neural cells, GABAergic neural cells, and non-neural cells. And we identify about near 500,000 cis-regulated elements at that time. So today's study, we extend the kind of sample to the whole mouse brain. You can see we have in total have 18 dissections from the anterior to the posterior for the mouse brain. And for each slice, we can have further detailed slice dissections for those regions. So here you can see the ABC part that corresponding to the detailed regions. And we call this, each part we call this one dissection. And for one dissection, we typically have 2 replicates. And those for one replicates, they are typically collected from at least 2 mouse brain. And for some particular subcortical region, if they are particularly small, we will collect more mouse brain to in order to get enough cells for our later analysis. And finally, so here I want to show you that the data we have compared with our previous study. So in total, we have now 2.3 million cells, and we have 117 dissections across the whole mouse brain, and also cover the majority, most of the major regions here. So the single cell ATAC data analysis is quite different compared with single cell RNA data. It's very sparse because we detect the signals on the DNA level and check if one particular DNA region is open or not by your manually modified team file, which it carries a pre-designed primer and can insert the corresponding open chromatin part. Well, the open chromatin here, that means there is no protein binding, or the DNA itself are not binding to the histone histones. So typically you can see the insertion within the two nucleosome, which typically will have the loss within about 100 base pair or less than 100 base pairs. You can also see some fragments well across multiple nucleosomes. And people use two different strategies to describe or represent the cells for the single cell text data set. One using the fragment base that tracking the fragment. So using the fragment itself to represent the cells. And basically in this strategy, we will partition the genome and check if some particular fragment will be located in the corresponding genome bin or genome part. Another way is we directly counting the insertion positions, like here. So given a specific peak, these two different representation will have different measurement accordingly. So here, at least the corresponding methods for different, while using different procedures described here. So SnapType is designed in our team by Rongxing, and our previous study is using the SnapType. And recently, Kai from our lab developed another version of SnapType 2. And we fixed some bugs, and the most important innovation in SnapType 2 is that we do a new kind of formulation for the representation. So that, and we also noted that we can do a very kind of a memory reduced way to do the eigenvector decomposition. This will be very important for our cell embedding. So in our previous paper or SnapType 1, we actually using the kind of based on the cell-by-cell similarity matrix and using the spectral embedding to do the low embedding or the low dimensional representation. But that's, you think about it, we have 2 million cells. So the 2 million multiplied by 2 million, this matrix is pretty huge. And later we noticed that actually we don't need the cell-by-cell symmetric matrix. We can directly using the cell-by-feature matrix, and we can using the particular method to do the same decomposition, so that we can now using all the cell together to do this accurately for the low dimensional embedding. So you can, and this package is written in Rust as a backend, and also use Python as the API. So you can, which is very easily to use. And you can see, compared with all the other methods, this method not only save lots of time, but also save lots of memory. And more importantly, based on several benchmark data sets, we noted that this SnapType 2 can keep the bioconservation here at a very high level compared with all the other methods. So in our data analysis in this time, we just using the SnapType 2 for our clustering and later analysis. So based on the SnapType in total, SnapType 2, in total, we have identified more than 1,400 cell subtypes across the whole mouse brain. So this figure, this is a UMAP showing using all the cells and show the low embed show the UMAP about the low embedding space for all these cells. So I use the different colors to label the corresponding class. Here is a non-neuronal cell major group, and this is glutamatergic major group, and also GABAergic major group. And then we will do the iterative clustering stage. So basically, we will pick up one cluster and do another round of clustering, and we repeat this kind of a clustering procedure several times, so that we can finally identify those large number of cell types across the whole mouse brain. A difficult part for this is trying to identify or do the annotation for the classes we have. And we do the annotation based on the single cell RNA data from Allen Group. So in total, they have 4 million single cell RNA data coming from 10x version 2 and version 3 technologies. And we're using the taxi data to simulate the gene expression, and then we can use the Surite to do the co-embedding and do the integration and transfer their labels to our data set. So in Allen's data, they in total have identified 5,000 clusters, and they organize the 5,000 clusters into a hierarchy structure. So at the very top level, they have 60 to 70 classes, and then they have about 300 classes, subclasses, and 1,000 the super tabs, and then 5,000 custom. So in this paper, we focus on our annotation or description, analysis on the subclass level defining island dataset. So here is the co-embedding space show that the two modality for the neurons can map really well, and also we do the consensus metrics for our data. So basically, the consensus metrics measurement, given a cluster in our data, we will see after transfer label how many cells are labeled to the same subclasses, and this can be the ratio for that particular classes. So you can see if the score tend to near the red part, that means that most of the cells in our cluster tend to have the same subclass labeled in island set. So you can see we have a nice consistent matrix listed here that mapping our more than 1,000 neuron cluster to 253 neural subclasses. This is how we do the annotations. And once we have the annotations, so then we'll identify the candidate cis-regular element per cluster. We do this by following a typical straightforward encode pipeline. Since we have replicates, we'll do the peak calling using the max tool with the replicates and pseudo replicates. And the filter only choose the conservative peaks, and also we have single cell level filtering based on the fraction of accessibility for the particular peaks. And finally, we have identified 1 million cis-regular elements. Here is corresponding their annotation in the mouse genome. So you can see a larger group of them are mapped to the intron region or intergenic region. And also there are some cis-regular element mapped to the corresponding transposable element like LP, line, sign here. And we do the overlapping with the encode this side, and we can see that 40% of our cis-regular element haven't been identified by their bulk level studies. So here, in order to further validate our candidate cis-regular regulatory elements, we check in their corresponding DNA methylation state. So those data can be shared by Akers group. So here you can see for the left part, this heat map, each row corresponding to one subclass, and each column is corresponding to one representative cis-regular element. And we do the kind of model analysis so that we can, group the peaks so that CCRE, so that they can have the tend to have the module patterns so that it can be easily observed in this heat map. So you can see, if you can notice that there are actually some cis-regular elements show a global chromatin accessibility across all the subclasses. And those are many such kind of conservative cis-regular elements, such as CTCF and so on and so forth. We also have a lot of subclass specific cis-regular elements. And on the right part, this actually corresponding to the DNA methylation state for the particular cis-regular element we have. And the lower or the dark color means that they are hypomethylation, and which I've shown previous that the hypomethylation signal is also an indicator for the chromatin for the cis-regular element. So you can see they actually map to each other very well. And we also checking the corresponding histone, the kind of chromatin accessibility and their DNA methylation state for the non-overlapped part, the non-overlapped cis-regular elements. And you can see still they have the very good alignment for the candidate cis-regular element we have identified. Okay, so now in the next stage, once we have identified this a large pool of cis-regular elements, then we will naturally tend to see how we can we identify or inference of the gene regulation programs. So we typically do this kind of inference in two ways. One is a very classical way to do the correlation analysis between the our cis-regular element and the downstream potential target genes, and checking if the chromatin accessibility and the RNA expression or the gene expression are matched to each other, or they have a higher correlation across the subclasses. And another part, we're using the latest, there are lots of methods talking about the gene regulation network, such as cell oracle or synaptic plots and so on. So the basic idea for this part is actually different compared with the left one, that they only consider the transcript factor. So basically, for example, we have the cis-directed elements, so you're mapping those cis-directed elements based on the motif analysis, and so get the corresponding transcript factor, and then they will later using for a gene and the transcript factor surrounding that gene, and focus on the single cell RNA-seq data and do the correlation or regression analysis to identify if some transcript factor can predict the corresponding gene expression patterns. And this kind of regression parameters can be used for the network. The positive means the TF potentially have the positive regulation for the corresponding gene, or they can be negative. And also this network have the weights. So we try both of the method in our paper. For the left part, we actually, so this also need us do the integration analysis from our taxi data and also the corresponding RNA-seq data. And once we have the integrated some subclasses level, so we can do the correlation analysis and identify, for example, here we only focus on the positive correlation pairs. We call this kind of a, how can we say, we call this kind of proximal distal connections. So proximal means the elements located in the cis-regular elements in the promoter regions. And distal means the cis-regular element not located in the promoter region, but within 500 case pair for this particular promoter. So in total, we have identified about 600,000 this kind of positive pairs. And this kind of cis-regular elements can be the potential enhancers in our later analysis. So here you can see that this part, I show you the corresponding heat map about the chromatin accessibility for all the potential identified enhancers. And on the red part, these are the corresponding downstream target of their, and this figure shows the corresponding gene expression patterns. So each row here is actually corresponding one particular cis-regular element in the left, and each row here is corresponding to the potential target genes they have. So you can see by checking each row, you can see they actually have a very consistent correlations or consistent patterns between each other. Here I will show you an example of a genomic track for the transposable elements region. So this is a transposable element, and this is a corresponding attack signal for this transposable element. They are, well, have very high open chromatin states, and the downstream, their positive PDC, which this part is corresponding to the downstream potential genes, and this the ataxic signal for this I think this kind of TSS related TSS regions, and also this is corresponding RNA-seq signal for the particular genes. So this is an example about the identified enhanced gene pairs in this paper. And another way we do we're using the gene regulation program to identify the corresponding transcript factor and their downstream genes. So we do this kind of way by using the cell oracle. So typically we will get for each subclass, we will get the corresponding network. And you can see we have the transcript factor and the regulation between them. And we can also check in the degree about the network. And the most important part, I think, is here, the heat map I show here. So since each TF, since for each subclass, we have, so in total in this program, we have used about 200 transcript factors. And for each TF, which showed here in each row, we can identify their important role in the gene regulation programs or gene regulatory network in one particular subclass. Here is in the columnar part. And so in this way, we can plot importance of the TF in a subclass level. So you can see here, some transcript factors show that their important role across multiple subclasses, and some transcript factors have only show their importance in particular subclasses. And we do this. We also compare our result with Allen's analysis. So in Allen's paper, they only use the single cell RNA-seq data, trying to identify the important transcription factors that define the corresponding subclasses. So the story here is that they for their clustering pipeline, they have 5,000 clusters, and once they organize all these clusters hierarchically, they're actually using about 200 transcription factors, and they are checking those states for different subclasses. They call it kind of potential regulatory code. So you can see from the left our data and the right Allen's RNA data. We also have the same order for the transcription factor, but we have due to the motif analysis, so we don't have all the transcription factors in Allen data set. And we also keep the same column based on Allen data set. So the pattern seems very similar between each other. But I think our advantage is that we integrate both the DNA level signals and also the corresponding RNA level signals to provide this a global view of this gene regulation programs in a cell type specific way. So now I've already introduced the how we identify the potential enhancer gene pair and gene regulation program. So next part, I will introduce about the conservation of the mouse brain cis-regulated elements compared with the human brain. So in our group, we also have a science paper released last year that talk about we probably have 3 or 4 human samples, and we also do the whole human brain ataxic analysis. So we can actually mapping our cis-regulated element to the corresponding human genome, and also checking if our cis-regulatory elements can map to the corresponding cis-regulated elements defined in the human genome. So you can see on the left part, so about 60% of our cis-regulated element can map to the human genome. And for those mapped part, 36% of those cis-regulated elements in our group, in our data, can map to at least one cis-regulated element in human data. And when we check the ratio of this kind of conservation of our data, we noted that there are a large group of small specific part cis-regulated elements located actually in the transposable element regions. And for the transposable element, here I want to introduce a little bit about the background. They actually are the mobile sequences in our genome. And for example, in human genome, more than 50% of our genome actually are the transposable element. And this transposable element have lots of copies in our genome. One important, for example, the most enriched part is the allele element. They have about 1 million copies in our genome. And typically for allele, it has 300 base pairs. And it's actually capture 10% of the human genome. So the transposable element, they have two mechanisms to replicate themselves. One is and then they have 2 classes. One is the retrotransposon, one is the DNA transposon, and they are very important actually to maintain the chromosome organization. For example, majority of the CT cell binding state, which used to identify the TAD boundary. The TAD here corresponding to the topologically associating domains, which is typically defined the chromatin loops. And the people recent studies say that within the chromatin loop, the cis-regulation typically will take their function to regulate the corresponding genes. And so this kind of TAD boundary are mainly dependent by the insulator, and lots of this kind of boundary part is derived from the transposable elements. So they can maintain the hair order structure, and they can also take a kind of function like transposable enhancer binding by the corresponding regulated elements and target the corresponding gene. This kind of if within TAD we see this kind of short range interactions, they can even across the long range interaction to regulate these genes. And a larger group of the transposable elements are located in the heterochromatin regions. So those regions typically will have a low activity or low open state for open state. And the transposable elements play essential role for those regions. And usually they have a high hypermethylation state and to maintain the heterochromatin stability. So there, I think you may hear this research that, so for example, this allyl element actually have a very interesting function that can lead us to absence of a tail by having a pair with another allyl element in the intern regions, so that it can introduce a specific gene splicing mechanism that can lead this kind of absence of tail. And also another, they also actually have a lots of function for the brain system. For example, here is a recent study showing that there also are retromalin. This is also another retrotransposon element, and they noted that this actually very important to help the oligodendrocyte from immature state to mature state, especially for the myelin part. So this myelin can wrap around the axons, can speed up the signal transduction, also support the survival of the neural cells. So it's actually this oligodendrocyte are non-neural cells and have a very important biological function. And this is transposal elements once they have the corresponding RNA sequences, they will bind into the particular proteins, and together they actually regulate the downstream genes. So now let's go back to our analysis. So we actually noted that there are a group of transposable elements here show very kind of a paired open complement state in cerebral regions, especially in a group of glutamatergic neuronal cells. If we look at the corresponding geoterm analysis, so how we do the geoterm analysis, because is that we have the enhancer promoter connections previously we have inferred. So we use the corresponding genes, downstream genes, to do the geoterm analysis. We can note that they actually show very significant glutamatergic neuronal cell activities. And here we list the top-ranked transposable elements and the corresponding downstream genes that play important role, well, at least show very high open complement state in our data analysis. So this may indicate that this kind of transposable elements may also have important functions, not only in non-neural cell, but also in a group of neuronal cells, especially it's kind of glutamatergic neuronal cells, which are many excitatory neuronal cells in our cerebral regions. So lastly, I want to introduce the analysis about deep learning with the raw DNA sequence we have. So the reason we do this actually inspired by an early paper, this informer. So basically, the informer structure is a deep learning method that can, using the raw DNA sequences listed here, so typically can have maybe 200 kilobase pairs as the input. This raw DNA sequence input can finally predict the corresponding tracks, genomic tracks. So these tracks can be either gene expression patterns, can be ataxic signals, can be chip-seq signals, and so on and so forth. With this kind of model, they actually found that this model can use to validate the potential enhancer state. For example, shown here, they focus on the particular gene listed here, and they using the experiment. So the CRISPR experiment validated the corresponding enhancers, mainly colored by the black color listed here, and in their model, they can calculate the importance of each base pair for this particular activity of this gene. And so you can see the hair scroll part corresponding to the major regions that have more important for the prediction of the gene expression for this particular gene. And those, you can see that the overlapping part is indeed the corresponding enhancer state. And another part is actually this model can do the in silico permutation study. So for example, they can permute the sequences, and also the permutated sequence means that for each base pair, they can they can change it to the alternative allele, and they can measurement the output. The output based on the, well, if the target gene have a overexpression or lower expression, and they can identify in this way, they can using the changes of the output to define the importance of the particular base pairs. They can, so in this part, they can identify the potential transcript factor binding state. So based on this interesting model, so we want to see if we can use this model in our data set. So we do this part. I think at that time there is no such large scale single cell data have been released. So there are deep learning model mainly based on the bulk level study, and here we apply this to our single cell level study. But this is kind of a suitable. But since we have a large scale of the single cell data, we can have lots of different subtypes. So that we can extremely extend this method to our brain system. So the input again are also the raw sequences, and the output will be the corresponding ataxic signals, not for one cell type, but for all the for more than 200 cell subclasses we have. So you can see this model can predict cell subclasses specific signals. And here, I will show you one figure here. So this corresponding to the tracks. So the red one corresponding to the one the model predict. The black one corresponding to the data we have. You can see the model can indeed predict the tracks very well, and also cell type specifically. For example, this global signals the model can predict, and there are also some cell subclass specific signals the model can also work in this way. And we then directly apply the model to the human brain data set without any further training. Since because we have the human brain ataxia data described previously, so we can test the performance of our data on the human brain data part. So I think the overall Pearson correlation is okay, but not that ideal. But still, based on the predicted track, you can see here, the predicted result seems okay, seems good. But we noted that we didn't have a good performance especially on the promoter regions. So this is our application about the deep learning models for our data set. So that's all the story I'm going to share for today's presentation. So here is the summary. Then I hope based on today's presentation, you agree with me that this is a comprehensive single cell analysis. So we have in total 2.3 nuclei, and we identified more than 1,400 cell subtypes, and we have 1 million cis-regulatory elements identified here. We do the enhancer target gene inference and also the gene regulatory programs inference. We identify the potential important role of transposable elements in the more cerebral regions for a group of excitatory neurons. And also the deep learning part indicates the conservation of the cis-regulatory element between mouse and human. Now I want to introduce about what we are doing. So at first I show you that the chromatin accessibility measured by the single cell ataxia data is kind of one part you can identify the cis-regulatory element, but there are lots of different kinds of cis-regulatory elements, right? For example, promoters, enhancers, insulators, and so on and so forth. So we can use a histone modification to help us to classify those cis-regulatory elements. In our lab, Chen Xu developed the corresponding technologies called paired tag, which can jointly observe the histone modification signal and also the corresponding RNA signals. And our colleague Johnny performed the paired tag sequencing technologies across the mouse brain. And here I want to show you our current analysis for the adult mouse brain using this kind of paired tag data. We have different kind of, so basically we have 4 different histone modifications, and we do the crossing and in total we have collected about at least 1 million cells and across major regions. So this is still ongoing work, and we are here trying to, based on the histone modification and the ataxia data we have, and also the single nuclear methylation data released in the BS project to identify or construct a real comprehensive epigenomic map for the adult mouse brain. And lastly, I want to note is that I want to add some comments, from a recent paper that they talk about the deep learning models used for identify the potential cis-regulatory elements. Their argument is that this potentially not enough for the only based on the genomic sequence, because they do the simulation study for the genome sequence and noted that if we randomly select the 200 kilobase per sequences from the human genome, we can identify lots of large number of the same 20 base pair regions for the 2000 kilobase sequences randomly generated from the genome, which indicate that so lots of the our genomes actually have a non-random replicate repeats, or some potential elements are also always code well in the same always show the code very similar frequency in the genome sequence. So based on the if we do the inference, we cannot distinguish if the red one or the blue one are the real causative part or the real cis-regulatory element. So he mentioned that based on the random experiment, we can overcome this problem. But this actually need us to do the synthetic DNA sequences and do the large scale permutation permutation study in experiment, and using those data further to identify help the model to learn the cis-regulatory element code. So that's all the content I want to show today. And thank you very much for giving me the chance to do the presentation. And I released the codes and also constraint data. You can check from this website. And this this project is supported by the many two NIH grants. And again, thank you very much. And I'm happy to answer your questions. And that's it. Thank you.
Thank you so much, Songpeng, for this amazing talk. If you have question, either you can add it on chat, or I think you can also ask directly to Dr. Songpeng. So before this start, people start asking their question. Let me ask.
Oh, I cannot see their questions actually. Yeah.
Oh yeah, I don't see the chat yet. There is any question. But Dr. Songpeng, like I know for you just showed that you can also look for the chromatin peak from the bulk DNA, and we spend so much time doing the doing the particular library for the attack-seq. So how how much confidently with your new algorithm or the existing algorithm do you think from the bulk DNA we could look at the peak?
Oh, I see. So I think the single nuclear attack technology is actually very popular recently. And I think previously people do the experiment that if you have for a cluster, if you have 500 cells, you actually can identify the peaks, recover at least 80% or 85% of the peaks identified using bulk level study. So I think this kind of single level attack-seq data actually, and also the method we are using, can indeed capture majority of the peaks. But you actually raised another good question that in our clustering pipeline, we actually have 1,500 clusters. So some clusters are larger than 500 cells in that particular cluster. I think we have more confidence for those clusters for the peak analysis. We do have a small part of the cluster have no more than 200 cells. So in that way, we also do the peak calling analysis, but that may lack of that confident, I think. But in general, attack signal is actually very kind of very strong signal compared with the histone modification we have observed. For histone modification, we indeed need lots of cells for the later analysis.
Thank you so much for your insight. It's really helpful.
Yeah, thank you.
Maybe I can ask one question. So very great work. I think you did a lot of work here. I have two questions here. One question is the enrichment analysis. Can you actually go to that slide, which you actually do the gene enrichment analysis? I was just wondering, like, how did they use the
This one?
maybe go this one, right? So how did you do the annotation here? Did you use any software to do the annotation and basically annotate to genes, right? And then do the enrichment analysis. So do you use any software or use your own algorithm to do that?
Yes, we actually based on our previous method here. So, sorry, here. So basically, we tracking, we use this kind of both the DNA data and RNA data we have, identify the potential pairs of enhancer and the gene. And so for the T part, we do so the T can have the overlapping with our cis-reactive elements. And once they have the overlap, we use the downstream target, the potential target for the transposable element. In that way, do the corresponding geoterm analysis.
Okay, so yeah, I think it's kind of similar as what we did when we try to annotate, so for example, the significant fragment when we do the ChIP-seq or ATAC-seq, so we use actually HOMA, so basically identify the nearest genes, right? Which genes actually.
Oh, I see.
So so this
we
is
also
kind of
actually try that. We also try. We also look at the closest gene, and we also using the genes inferred from this method. Yeah, because we have two modalities, so we can integrate them and see the correlation with them. So this is another level of indication. Yeah, but we also do the same thing as you described. Yeah.
Okay, great. And I also saw that the deep learning technology you actually use in this project is, the accuracy is pretty high, which is very interesting to us also. I was just wondering, did you actually apply any other, deep learning technologies, to be used in this project? What do you think about the deep learning, technology in the bioinformatics analysis for any future project? Do you have any recommendation or any suggestion here?
I think we try. So the deep learning part actually from another PD candidate student in our lab, Ethan. He did lots of modification for the percentage model. Percentage model is actually a very popular model for recent years, just like what I showed for Informer. And indeed, they actually perform very well, at least for the mouse data themselves. And I think now I'm actually pretty interested in I'm not actually so my kind of feeling is that or I have some concern about this part. So the basic aim for our deep learning model is trying to identify or trying to perform the in silico permanent study. I think that's the main goal for our deep learning model. Otherwise, we don't need to do the model prediction because we have the data. So currently, we can only verify the model performance based on the signal we have, but we didn't actually measurement if this simulation permutation study can indeed have a good performance. This is actually one part I think we need to pay more attention later for the deep learning studies. Another part is I think for the last slides I show about the recent comment from another paper for typically to argue that the kind of raw genome sequence used for deep learning model we cannot capture the real we cannot capture enough. We cannot predict the cis-regulatory elements well, and so I think in that case so there are lots of potential data leaks. So data leaks here is that we have, for example, we have a transposal element. They have lots of copies themselves, and currently for our training and testing data set, we just randomly partition the genome beans and select part of them to be the training data set and the rest of them to be the test data set as previous also the method do the same things. But since we have for example, the transport elements are shared between the training and the testing data set, so the potential data leaks may also lead our overestimate about the model performance. So that in that way, we actually need more attention on how we design the training and testing data set. And this, I think we need to do further after this work. And another part is So other part is, I think I'm also very interested in using different modalities. So here we're only using the attack signals. But if and now we can have more signal, for example, the methylation signals. I haven't used here. And also now we have the histone modification signal I haven't used here. So the model can indeed, I think the simple model as a modified model can do some modification and predict not only cell type specific one modality, but also multiple modalities. That may let the model really learn some biological insights, I think. So that's another dissection I'm pretty interested in. Yeah.
Great, thank you so much for sharing that.
Yeah.
Do we have any other question from the audience? All right, Sanjit, that seems like we don't have any more questions.
Yeah.
Maybe you can stop here.
Yeah. Okay. Should I stop sharing or?
Yeah. Thank you, everyone. Thank you for, for the great presentation. Great work, and thank you everyone for joining us. So have a great day.